Hi, everybody. For those who don't know me, my name is Jason Kelly. I am the Applications Director with Lighthouse Worldwide Solutions. I'm pretty active on LinkedIn. I do a lot of posts, so there's a QR code there if you want to get in touch with me. And we also have a knowledge center on our website that's really informative. There's another QR code there where you can go to our knowledge center and type in multiple topics on environmental monitoring clean rooms, anything to do with standards, best practices. We have a lot of good tutorials, and it's all about getting the knowledge out to use to empower you to be the smartest person in the room. So let's talk about our subject today. And I wanted just to talk about a little bit about aseptic processing. The art of performing a specific process within an ultra clean environment to ensure that the product, in this case, an injectable, is transported from bulk sterile containers and it's filled into vials or syringes or capsules. I'm going to take a deep dive into continuous monitoring of the aseptic processes, and we will concentrate on microbial monitoring, especially since EU GMP Annex 1's latest release has pushed us further into that direction where continuous microbial is now required. So the expectation for continuous microbial monitoring in critical environments where container and product storage is at its highest risk. Um, for instance, like these containers, they're filled and capped in a sterile environment, typically ISO 5, grade A. The environment is monitored continuously during the aseptic processing for any particular contamination using a combination of particle counters and air sampling, active air sampling, and settled plates. And the goal of the operation is to prevent contamination of the product and that may affect the quality and sterility of the parental product that's been injected directly into the patient. So they do say that continuous monitoring is more than just a guideline, it's a safeguard against contamination. At the end of the day, these are the patients, and we really need to be mindful that the product is going into a patient who has probably already a compromised immune system. So that product needs to be safe and effective. So what does EU GMP Annex 1 guide us on? So Annex 1 provides a general guidance that should be used in the design and control of facilities, equipment, systems, and procedures used for the manufacture of all sterile products, applying the principles of quality risk management, QRM, to ensure that microbial, particulate, and endotoxin pyrogen contamination is prevented in the final product. Now, when we look at, a contamination control strategy, we know it's a comprehensive approach designed to prevent, reduce, and manage contamination in these environments where cleanliness and sterility is crucial. And the strategy encompasses various methods, protocols, and procedures aimed at controlling the introduction, generation, and retention of contaminants in these critical environments. And the contamination control strategy, we're looking at a whole governance of your facility, starting with the risk assessment. We also want to raise personal awareness and have a quality culture within the environment. That goes down to training, hygiene, gowning, the actual process design itself, the selection of materials, the equipment design, the cleaning, the sanitization, and the utility design and cleaning and sanitization for those. So these are the contamination controls. The contamination control strategy then talks about the validation of these controls. You want to validate the personnel, to requalify them. You want to validate the processes and the life cycle of those processes. We want to implement analytical methods into the life cycle, look at the facility equipment and qualification. And then there's also the monitoring of the controls, personnel monitoring, in-process monitoring, monitoring materials, environmental monitoring, utility monitoring, even down to pest control. So the contamination control strategy is a total governance of the whole facility, and it's a multi-department collaboration. And that collaboration will come about all of the data that's being put into the contamination control database. So we look at, what is the strategy? Well, it's about risk. It's about a risk assessment. The process begins with a thorough risk assessment to identify potential sources of contamination, assess their impact on the environment, products, and processes. And this includes evaluating factors like raw materials, equipment, personnel, and the manufacturing process itself. So you're going to be looking at everything that's brought into the facility, the actions within the facility. If you're looking at the room, you're looking at traffic flow, product flow, all that sort of stuff. And it's put into a risk assessment. I'd recommend ICHQ9. Follow that guide on risk assessments. I do have separate webinars on how to develop a risk assessment in our past webinars. You may want to jump on and have a look at some of them. But it is a holistic approach. And then you want to establish what is your contamination control strategy. Well, you want to establish the control measures. You want to conduct regular risk assessments, implement robust policies and procedures, utilizing technology solutions, and effective communication challenge. So you want to have all this process in the design of your clean room, the control of your environments, implementing strict access controls and personal hygiene protocols, even the awareness of clean room behavior, how to act in a clean room, what to do, what not to do, using appropriate cleaning and disinfection procedures, establishing material handling protocols. So what you're bringing in to the clean room is really important. So these are all part of your strategy, including the monitoring and testing. So you need to have regular monitoring and testing of the critical environments. These involve continuous or periodic testing of air quality, surface cleanliness, water purity, and other environmental parameters. The use of monitoring equipment and routine sampling helps detect any deviation from established cleanliness standards or established baselines. It's important to have a good contamination control environmental monitoring plan where you'll use multiple instruments and tools to do your environmental monitoring. Particle counters will just be part of that. Alongside with that is the training and awareness. Training the personnel on the equipment, on the processes, on themselves, on the proper aseptic counting up the technologies that they're using. And it all will help with developing a more robust contamination control strategy. Documentation and record keeping. You want to maintain detailed records of contamination control activities, monitor your results, and any corrective actions that are undertaken. You need to assess the effectiveness of your contamination control strategy, also for regulatory compliance, and looking at your baselines and strategizing your alarm limits based on your environment and your conditions. And that also leads down to continuous improvement. So the contamination control strategy, it's not static. It requires regular review and updating based on the monitoring data, process changes, any new regulatory requirements, and technical advancements. Anything that's out there that's new that you may implement that would bring about more data sets and possibly better data. And continuous improvement efforts aim to enhance the effectiveness of the contamination control measures. Another important factor is the emergency response and the corrective actions. And the strategy should include procedures for responding to contamination events. How do you respond to a particular event? That's really important. And what do you implement and what corrective actions are used to prevent reoccurrence? So in summary, a contamination control strategy is a systematic and comprehensive approach that integrates various practices, procedures, and controls to manage contamination risks in sensitive environments, ensuring product safety, quality, and compliance with regulatory standards. And we need to look at what actually are we monitoring. So let's look at the average human hair, for example. It's about 100 microns. Some people have thicker hair, some people have thinner hair, but we take an average of 100. Like we're getting down to sizes of bacteria at 5 microns. And if we were to look at trying to look at a bacterial particle, it's going to be very difficult. It's going to be impossible for the naked eye. So we're using technology to detect particles that we can't see, using particle counters for detection, and we were using air samplers to pull in the sample and see what incubates, if there's any CFUs. So it's important to understand we're definitely looking at, or not looking, but we're using instrumentation that will look for us to monitor the different types of contamination that could be in the clean room. Like just to put things into its perspective, like in the semiconductor industry, they're down to 3 nanometer line widths. And how many 3 nanometer particles would fit across 100 diameter, a hair of a diameter of 100 microns. You're looking at about 33,000 3 nanometer particles would fit across that hair. So when we get down into the semiconductor industry, it's even more important controlling the environment and making sure that those particles aren't present. And it's a challenge because as ourselves as the main contributors to contamination in the clean room, we're present. But the trends are moving towards moving the contaminators, the people out of the clean room. So continuous monitoring, it's nothing new in EU GMP Annex 1, and the requirement goes far back as 2003. We see that in 2003, the guidelines were for particulates using particle counters. And now we look at the latest guide, and it talks about, this is still looking at the 2003, it talks about monitoring should be frequent. Where most companies would do a pre and a post with their microbial sampling. So they'd have a pre-post data. Monitoring continuously during the process does cause some challenges with the technology. And you also want to limit the frequency that you're changing out your media dishes as well. So the new standard with EU GMP, it did section 924 talk about continuous avoidable error monitoring in grade A, and a similar approach considered for grade B. So they're pushing us in this direction of continuous microbial monitoring, and it's up to us to react and to try and work out the best way to monitor. And the tools that we have at the moment, we have the air sample settle plates, you have the active air samplers, contact plates are used as well. So we do have these multiple tools. There's also another part in the EU GMP Annex 1 that talks about using any new technologies, but also that you need to also understand the procedures that you're using, and what results you're getting, and the efficiency of the sampling methods. So there's a lot of information out there, and like for example, you could consult with ISO 14698, which talks a lot about the selection of the active air sampling devices. There's quite a lot of information in there. These are some of the aspects of 14698. We do have EN 17141, and also there's going to be an update. ISO 14644 Part 20 is actually, we're going to start reviewing that standard, and maybe in 2 years that will be updated for the new microbial standard. But things like looking at your D50 of your air sampling device, looking at make sure you got a HEPA filter on the exhaust if you're using a device that has an exhaust. You need to get a size resolution down to 1 micron, and then looking at your biological and physical efficiencies. So it all adds up, and there is a section in there, as I said, in 14698 about selecting the appropriate device and making sure it's fit for purpose. EN 17141 talks about the establishment of microbial control. It's another good standard to consult. This one came out in 2020. Identify sources of microbial contamination, assess sources and routes, risks, and then address those risks. Establish a schedule for monitoring microbial contamination, including valid sampling methods. Establish appropriate alert and action levels. So the list goes on and on. And as I said, we do have the update happening. It's just in preparatory stage here in the life cycle of that document, and I do think it might take about 2 years. I am a member of the ICCS who write these standards, and we are actively looking at these standards and updating and getting feedback from industry. So the tools that we do have, as I said, we have the air sampler, the settle plates, contact plates, the particle counters. They all have good. They give you good data sets. The contact plates either on surfaces or on the actual operator in the room that's providing or working on the aseptic process. Glove prints. We do have the also settle plate and air sampler data that will come in. So these are tools for microbiological monitoring, and these are important to use multiple tool sets, so you're getting multiple data sets back. For example, you see those tools in action, the active air sampler in a filling line and the settle plate also in the filling line. So you're getting multiple data sets, and that's what they would look like in that operation. You can also see here circle as a sample probe for a particle counter. So we're making sure we're getting clean air sweeping over that process. and ensuring that that environment is an ISO 5 grade A environment. Again, looking at the types of instruments, you do have the portable or continuous sampling. The portable units can be brought in, used effectively, or you could have a continuous system that's set up to a continuous vacuum, connected to software. The operator can start and stop remotely or start and stop locally, and add or replace the media once it's expired. And this is what these devices look like. In terms of using the monitoring tools, if we take a biological safety cabinet, for example, we implement the particle counter here. We have a remote particle counter. I like using these because they have a small footprint and hey, they can be put inside the cabinet. That also eliminates the risk of particle losses in the sampling tubing. There is a new guideline, a technical report that came out. It's ISO/TR 14644 part 21, and that discusses validation of the particle losses in the sampling system tubing. And there's a whole webinar on that topic. We did have some input as a company into the development of that technical report. And we have an applications page on our main website. You can scroll down and see how it's done. We actually also have some tutorial coming out on how to validate the particle losses, so keep your eyes open for that one. Settle plates, implementing the settle plates into your biological safety cabinet. You see they're positioned at the work level. And then you have the active air sampling device, again, a remote active air sampling device that will connect into a vacuum system. Again, these are monitoring the process. Your particle counter is ensuring you have that clean air from your HEPA filters passing over the process. That's how I would go about monitoring and implementing these monitoring tools. You see you have the critical zone here where the operator will conduct their aseptic processing techniques. They could be making the aseptic connections, or they can be doing all sorts of activity that requires that environment to be monitored for sterility and to ensure that the operator is not transmitting any contamination. By the actual technology of the biological safety cabinet, you'll have a window that comes down with a sash window that's adjustable, but you'll also have the ability to stick your arms in. There's also a positive barrier developed with the airflow coming down, so it's stopping any contamination from the environment outside of the cabinet getting in, and it's also preventing anything from that critical environment getting out. So there's two methods of activity going on where the operator is being protected and the product's being protected from the environment. Looking at the viable air sampling technology, I do think it's important to understand how it works. So you do select the right model for your application. Typically, these have a sample head. You're going to put your agar media inside, and is a base plate. And there's we're going to talk a little bit more about the technology. So it's important the distance between your sample head and the media. There's a factor called the D50, which I'll get into. The D50 essentially is the resolution of the air sampler. It's the capability of collecting the smallest size. And in ISO 14698, it talks about a D50 of about 1 micron. EN 17141 talks about a D50 being less than 2 microns. So you can see how this actually works. You got the suction system, the air is coming sampled in. You can see particles that have a smaller size will impact easily on the media. Same with the particles. The particles of the larger size will impact more easily because of their inertia. The particles that have a smaller size, and this is the D50 in action here, you'll see there's a probability of 50% probability that that particle will either impact on the media or it'll be taken away by the airstream. And then that effectively is your D50. So that size, the standard is saying the D50 must be at least 1 micron, particularly when you consult with 14698. So that's a little bit on the technology. The D50, the collection efficiency, as I said, it's the probability of 50% of that particle landing on the media or being swept away. And that contributes to the collection efficiency and the resolution of the air sampler. And believe me, there's some air samplers out there with a D50 of 10 microns, meaning that they do not have the ability to detect any particles or pick up any particles below 10 microns because they'll be swept away by the airstream. So again, you need to look out for that and follow 14698 as a guideline for selecting an active air sampler. I strongly recommend that. I also would say low flow is the way to go, and particularly with this continuous microbial sampling, and if you're using either a portable or a remote sample head, low flow, go for something that has a lower flow rate, because you understand that's going to implement the number of times that you're swapping out your media dish. If we take an example here, if you have 100 liters a minute, let's say we're working in a 8 hour environment where we are aseptically processing for 8 hours and we're doing that 7 days a week. Well, if you're changing every 10 minutes because the flow rate is so high, you're going to be going through about 17,000 media dishes a year, and you can see the cost line up. If we reduce that flow rate down to 25 liters a minute, there's still significant changes being made, and you're changing out your media every 40 minutes. You're going through 12 media dishes every 8 hours, 84 per week. There's a lot of evaluation, incubate those, evaluate them. So there's a lot of work going on. Typically, if you get down to 10 liters a minute with your flow rate, then you can reduce the number of media dishes to something more manageable. And also with that, the cost will be reduced as well. So that will definitely help you establish um, understanding of the flow rates with your active air samplers. Um, there's also rapid microbial tools out there. And it does say in the Annex 1, the adoption of suitable alternative monitoring systems, such as rapid methods, should be considered by manufacturers in order to expedite the detection of microbial contamination. Now, there's two methods. There's the in situ active biological monitoring using biofluorescence technology with these particle counters that have sensors that can detect biofluorescence from the particles that will have microorganisms. And there's also the rapid sampling. So once you've taken your sample with your active air sample, you can have that incubated by a rapid sampling system that also uses biofluorescence, but it's also using magnification and digital cameras and able to detect CFUs forming that are not naked to the visible eye. So that they reduce the actual incubation periods and the times to identify CFUs down from 72 hours, sometimes down to 4 hours, depending on what you're looking for. So I would say just to consider these technologies, and I will go through those technologies in a little bit of detail. But at the end of the day, everything's based on vibration. This fluorescence technology is based on vibration. And Nikola Tesla, he said, if you want to find the secrets of the universe, think in terms of energy, frequency, and vibration. And that's how the technology works. The biofluorescence, it's been around for a long time, and the use of biofluorescence to detect microorganisms became more prominent with the development of the green fluorescent protein GFP technology in the late 20th century. GFP, initially isolated from jellyfish, was recognized for its potential in biological research in the 1990s. And the technology advanced rapidly, allowing researchers to express GFP in virtually all organisms under a study within a few years. So, the way the technology works, it's about how excited you can get the cells. Um, so, the detection of viable particles from an air sample stream or from an exposed media dish using ultraviolet light wavelengths, typically of about 480 nanometers. The cells will pass through these wavelengths, and that wavelength will excite the cells, and they'll actually give off a biofluorescence of their own, a visible green light spectrum about 510 nanometers. And this is what the biofluorescent particle counter will pick up. So that technology is around. I have been around this technology for about 12 years. For the rapid microbial detection, similar, it's using an autofluorescence, which is replacing the human eye. And they even have advanced robotic tools to work their way through multiple samples and increase the actual efficiency. And a company called Rapid Micro Biosystems develops this. I have no affiliation with it, but the technology is pretty straightforward. And it's basically absorption and re-emission of light from living organisms, and talks about the present your sample, exposure to light, the emissions of the fluorescent, the detection, the analysis, and application. I won't go into too much detail, but the bottom line is this detection enables a lot more faster detection rates compared to traditional methods. You can see on the graph here, traditional manual methods in blue, and the rapid detection with the growth direct from this particular company here has enabled faster detection. And again, as soon as you have faster detection, you have quicker information about your process to make more informative decisions. Then also the other start is the biofluorescence technology, the in situ monitoring. There are a couple of products out there. MicronView is one, TSI BioTrack is another. Are they going to replace traditional methods? Well, that's the question. Again, you need to look into the technology. One of these products, the TSI BioTrack, actually is able to sample the obviously, it's going to sample the air and it's going to give you a biofluorescence hit if it's present or not. But they're also capturing that sample onto a gelatin membrane filter. There's a couple of things I'm curious about. Is that it's only 1/10 of that sample is actually being captured. But there's also some false positives, about 7% false positives with the technology. But at the end of the day, whether you deploy this technology or not, you're still going to have to wait up to the traditional time of 72 hours. And it's the same with traditional marketing. You're still waiting for your CFU growth on your media dishes, and your particle counter is still going to act as the flag. So just be aware of the technology and what it represents. You're still playing that waiting game. And also, there's some issues around. I've been around this gelatin filter technology for quite a while. I was first introduced to it 20 years ago with the Sartorius who developed the gelatin membrane filter with their air sampler. So there are extra steps involved that can introduce more false positives to the sample to be aware of. And looking at the particular unit here, it's a big beast. This BioTrack is pretty big. And then trying to get this into in situ areas where you want to do continuous monitoring, I don't know that's going to be feasible compared to the traditional methods. You've also got to contend with the problem here. You want to monitor a filling line. The traditional methods are fitting in over here in this space. They're quite small, easy to implement, but you've got put this unit outside in a grade B or C area, and then you're sampling inside a grade A. Now, my question is that feasible? Well, you're going to be loading your gelatin membrane filter in a grade C environment, and to me, it's not really feasible. In fact, I know an auditor in the UK recently stood up in a meeting. It was at a PHSS event that you guys had recently, and questioned this. But I do see a place for this technology. I don't see it in this traditional continuous monitoring environment, but it is pretty good for troubleshooting. How I would implement them? Yeah, that's a good question. Yeah, you have your traditional method. I'd still stick with the traditional method. The biofluorescence technology is good. It's exciting. It could be implemented as a troubleshooting tool. It could be implemented in isolators away from human activity as long as you're loading your sample media in the same environment that you're testing, same grade of environment that you're testing. So it may mean purchasing a lab cabinet, a small lab cabinet to put it into. And then you've also got to contend with the particle losses in sample tubing if your tubing is rounded too long because of the size of the equipment. The MicronView model down here, it's smaller, but it doesn't capture the actual sample. So again, you've still got the issues with false positives to contend with. Something that you can live with or not, I'm not sure if it is, particularly when you're looking for zero CFUs in your process. So looking at the implementation of traditional new, we take a filling line, for example, we follow the path of the vial, comes out of the sterilization oven. So we have an open vial, goes into the accumulator here, and then it'll find its way to the fill line, onto the fill head, and then onto the stopper and capping stages of the process. So each stage you want to be monitoring traditionally particle counters, air samplers in these settle plates in these areas where you can have coverage to make sure you've got enough coverage as the journey of the exposed vial goes through its process in the filling machine. And again, these are some of the locations with air sampling devices, these remote active air samplers, just highlighting them. And we also have some particle counters there. Looking at the implementation, I will include obviously settle plates, which I've included in this drawing here, you can see, and the active air samplers as well. So you have the trifecta approach here with 3 different data sets. And if you do have an event along the way, well, then you can say over this area here, you have a high count. Then you can implement your troubleshooting tool, your biological sampler to see if it's got a trend of biological events that will tell you you've got some issue in that area and you need to find the root cause. So looking at, you know, Pharma 4.0, we're moving on because they do like the contamination control strategy, Pharma 4.0, they all go hand in hand. We look at, um, Pharma 4.0, it's aligned with Industry 4.0. And yeah, I do like to say my superpower is being contamination control and expertise. But getting it back to, you know, what's your superpower? I think we all should be looking at these advances in technology, and Pharma 4.0 is tailored to meet the pharmaceutical industry's unique challenges and strict regulations. And it's about leveraging digital advancements to boost drug manufacturing efficiency and quality, insurance, compliance, and fostering innovation. And I said these biofluorescence tools, they do have a place as one of these tools to help you along your journey in ensuring the product safety. And again, we need to look at the technology and what it's about. And the core, Pharma 4.0, it's redefining the industry by digitalizing data and the processes, linking information, fostering transparency, and enhancing adaptability. This interconnectedness leads to swifter decision making, as I said, better control, and superior product quality. It's a holistic approach that binds technology with the processes and people, revolutionizing the production landscape. So, at the heart of 4.0 lies the principle of holistic control strategy lifecycle management, underpinned by data integrity by design. Achieving this requires an organization's digital maturity within the Industry 4.0 framework. So your facility, your organization needs to have a level of digital maturity to implement this. And it's a critical component of this new area in continuous manufacturing, which depends on a constant stream of data from environmental monitoring systems, ensuring digital regulatory compliance and traceabilities. And key aspects of Pharma 4.0 include the digitalization of operations, the data integration of and the analytics, smart manufacturing, artificial intelligence and machine learning, advanced robotics and automation, continuous manufacturing, regulatory compliance, even down to supply chain optimization, and even looking at personalized medicine, looking at the rise of ATMPs and those, personal medications that are being developed in that space. And as I said, the digital maturity and the data integrity are key to realizing the full potential of Pharma 4.0. And some of the aspects, as I mentioned, digital operations, implementing digital tools and solutions across the manufacturing process, from research and development to packaging and distribution, leveraging big data and advanced analytics to gain insight that drive better decision making and predictive maintenance, smarter manufacturing, using smart devices and the Internet of Things to monitor and control the manufacturing environment, ensuring product quality and operation efficiency. Let me give you an example of implementing this digital technology. We have a traditional particle counting air sampling system like we saw on the film machine a couple of slides back. What if we were to integrate digital cameras with facial recognition and body recognition in terms of the clean room activities? Let's say you get a particle spike on your particle counter. We could have these cameras zoom into that location and see exactly in real time what the activity may be causing that spike in particles. Was there an operator present? Did the operator intervene into the process? Are they aseptically gowned up? Is there face covering, their sleeves, preventing exposure of skin? Has the gloves got any holes or slashes in the gloves where, particles can be generated directly from the operator? And these are biological particles. So this technology can be implemented where you have facial recognition algorithms in the technology to look for these problems, alert straight away. So you're getting down to your root cause analysis really, really quickly. And again, using advanced robotics and automation for artificial intelligence and machine learning in this continuous manufacturing space, that will enable better regulatory compliance. So these are some of the aspects of Pharma 4.0. Now we get back and look at EU GMP and the latest revision. It's testament to the evolving landscape of aseptic processing. And it brings forward stringent requirements for environmental monitoring, aiming to safeguard the manufacturing of the sterile products. And it's a regulatory guideline. EU-GMP Annex 1, it's a regulatory guideline for the manufacture of sterile medicinal products, ensuring their quality and safety. And the contamination control strategy, it's an organized approach to maintaining a clean environment throughout the manufacturing process. It encompasses all aspects of production that could affect the sterility of the final product. This includes the design and maintenance of clean rooms, the handling of materials, personal hygiene, gowning, equipment sterilization, environmental monitoring, and process validation. And the latest version of this EU-GMP, it was released in 2022, and it came into approach in November of 2023. They've given an extension of November 2024 for separate RAB systems before that gets fully implemented. And in the context of Pharma 4.0, the digital data becomes increasingly important for the contamination control strategy that just outline these technologies that become available. And the use of digital technologies such as real-time environmental monitoring with particle counters, air samplers, and advanced data analytical tools allows for the collection and analysis of vast amounts of data. And the data can be used to make informed decisions quickly, improve the quality of the products, and ensure compliance with regulatory standards. And digital data also supports data integrity and governance within the contamination control strategy. It ensures that all data related to the contamination control is accurate, complete, and reliable throughout its life cycle. And this is essential for both regulatory compliance and ensuring the effectiveness of the contamination control strategy. With digital data, companies can adopt a more efficient, automated, and error-resistant process, reducing the risk of contamination and enhancing overall product quality. So continuous environmental monitoring is no longer optional, it's a necessity. By monitoring for viable and non-viable particles consistently, we're not just catching contamination events, we're preventing them. This proactive stance is crucial for patient safety and product quality. And continuous monitoring and continuous environmental monitoring is more than just a guidance, it's our safeguard against contamination. So in the world of Annex 1, digital data is king. The heart of Annex 1 compliance lies in digital data. And real-time monitoring through advanced devices like smarter, digitally advanced particle counters, air samplers connected to automated monitoring systems provides a data-driven foundation for environmental monitoring, ensuring that we meet the stringent Annex 1 regulations. And in the world of aseptic manufacturing, timing is critical. Digitalization is not just about adopting new technologies, it's about building sustainable compliance. And with enhanced digital technology and environmental monitoring, we create a contamination control strategy that's not just effective today, but is resilient for the challenges of tomorrow and beyond. And at the heart of Pharma 4.0 lies data integrity and governance. It's about ensuring the accuracy, completeness, and reliability of the data throughout its life cycle. Think of Alcoa, think of 21 CFR11, all those data integrity protocols. And so that this is what allows us to make decisions with confidence. Like, how do you know the data lifecycle from the collection system to the digital transfer has maintained integrity? I would say look at the technology from the EM monitoring vendors, what they're providing, and see how they manage data integrity in their technology. For example, how do they validate bad data? Does the actual particle counter have the ability to pick up any bad data? Well, some of them do. Some of the technologies have a lot of dynamic sensors inside that can pick up the possibility when a device is picking up bad data and acknowledge that it's bad data. And that will tell the user, listen, there's something going wrong with this sensor. We're not confident in the quality of the data. So that particular instrument can be put aside and brought out to service and not be continually used, where you send it on for calibration maybe 6 months, a year later, and you find it's failed the calibration. That's going to raise a lot more issues. So data integrity ensures that the environmental data recorded by particle counters and other monitoring devices and cleaners is reliable and it can be trusted. So the trifecta of environmental monitoring compliance I talk about a bit, looking at getting that data to where it needs to go. We do have these smart particle counters that can connect to LIMS systems, get into your contamination control strategy. And the digital age is simple. The manufacturing digitalization provides robust data governance, stringent contamination control. Together, they form an impenetrable shield against contamination risks by digitalizing EM data. The data becomes instant and available. And with the right technology embedded in your particle counter, for example, you can seamlessly integrate the EM data, the digital data, into a centralized database and use AI to help make real-time decisions using multiple data sets from the contamination control strategy. Because as you can imagine, facility-wide, all these data sets coming from all over different departments put into a centralized system where AI will start to look for trends and patterns within that data to help you perform better in your manufacturing of your aseptic product. Quality by design is reshaping the environmental monitoring. It's a strategy that doesn't just solve problems, it prevents them. By integrating QBD, we ensure that our clean spaces aren't just controlled, they're intelligently managed. And I've seen like now they're developing, or they have developed filling machines that are totally robotic. We're removing the operator totally out of the clean room and the aseptic environment. And these systems are out there. I was at an ISP event in Waterford in Ireland a couple of months back where they introduced some of this technology, and there's no human activity at all. It's all automated. Even the introduction of the materials and everything else that's used in the process is all automated. They're really interesting. So with QBD built into your EM systems, real-time environmental monitoring will revolutionize batch release processes. It enables us to respond immediately to data records, streamline product release, and enhance market readiness while upholding the highest quality standards. Continuous manufacturing will need robust environmental monitoring systems with reliable data to enable batch releases to be quicker and with confidence. And these monitoring systems, whether portable or fixed, need to integrate and have reliable connectivity to the main CCS database. So where's all this digital advancement leading us? You know, is this image the future of aseptic manufacturing? I don't think so. Um, I do think that we're going to have more robotics involved in the clean room, mobile, removing the mobile particle generators, us humans. Where are we going to go? Well, outside of the clean room, we can be involved in the analytics sides and making sure that all the processes are working correctly, the AI, the information, looking at all this data. So I do see a stream of operators continuously monitoring the data sets that are coming in. And then there's also the whole army of maintenance techs that will take care of all of these robotic equipment. I started my career off in Intel. I worked in Fab 10 in Ireland. We made the Pentiums and the machines, the wafer fabrication machines. They needed a huge crew of maintenance techs to maintain them continuously and have preventive maintenance to ensure that the uptime of those systems were at a maximum. And like I see the same happening here. Like the pharma industry will adopt some of the automation that the semiconductor has already embraced. So the future of this technology is lying in taking us out of the clean room and having more technological advancements in the analytics side of things. And at the end of the day, it's all about getting that data quickly to the correct decision makers, and therefore having the confidence in the product when that product is being shipped out. And as we've seen, the end users, they need to have that confidence that what's being injected in their bodies is going to help them and not kill them, to be frank. So, the integration of Pharma 4.0 into aseptic manufacturing paves the way for a future where efficiency, safety, and compliance are not just goals but guarantees. Again, if you do want to get in touch with me, please send your questions to me. I apologize for not being available in last minute emergency. But please send your request to me. My email is jasonk@golighthouse.com, or you can scan in the QR code and connect with me with LinkedIn and ask some questions. And I do have an appointment facility there where we can have a direct conversation if needed. So again, thank you very much. And I do hope you all have a great rest of your day. Take care.